Today we will discuss about how they characterize the clustered and single tumors and APC. So far we have understood that some of the tumors are present in the single form, that is they do not interact with any of the T cells, and similarly we have also seen the data that APCs are also exist in the single form and they are also clustered with the T cells. So in this video we will discuss, is there any difference in the single tumors and the clustered tumors, or the single APC and the clustered APC with T cells. So we will start with the tumor cells. So first what they did is they took the RNA-seq data of the tumors, and using the UMAP they are able to make different populations of the tumors that are exist. Then they characterize these populations using the publicly available RNA-seq data, and then by comparing the gene markers they are able to characterize which population belongs to which cell state, and they are able to find out different cell state. And thereafter, once they find out the different type of cell state that exist in the samples or in the patient samples, then they compare this cell state in the single tumors and also in the clustered tumors, and in that they are able to find out the two populations or two cell state that increased in the case of clustered tumors, that is immune cells, and the second one is this stress hypoxia state. So these two populations are increased in the tumors that are derived from the T cells cluster, that can be seen in the right hand side of the panel H. And from literature they are able to find out that this kind of tumor populations are known to attract T cells. So this could be one of the reasons why T cells are bound, T cells cluster with the tumors are more formed with this kind of cell state, that is immune response and the stress hypoxia conditions. Thereafter they did the similar analysis on the APCs also, so they are able to find out the different populations of the APCs that is shown in the panel I, and thereafter based on the data that is available publicly they are able to characterize these populations into different cell state based on the gene markers, and these cell state are shown in this panel J, where we can see that they are able to make many subpopulations or many cell state of the monocyte or macrophages from the different subpopulations that are identified using the UMAP. And here they also compared the or did the same analysis, that is they compare the different cell state in the single APCs and the APCs that are derived from the T cells cluster. Here they are able to see that here also they are able to see two populations are increased in the case where APCs are segregated from the T cells clusters, that is C16 high, that is monocyte like populations, and also C1q high, this C1q high, this is inflammatory macrophages like populations. And pardon for the pixelations, this is because I have zoomed in the image too much, as this is the third part of the figure 3. So let's come back to our discussions. So they are able to see that two populations are upregulated in the case of this APCs that are derived from T cell cluster. This also indicates, and when they check this kind of populations they also indicates the points that, or indicates towards that this kind of APCs are known to attract T cells, and this could be the reason why the T cell clusters are formed more on this kind of populations. So overall what they are able to conclude is that the T cells also has a preferential towards binding to the tumor's cell state, and also towards these APCs, and as this can be validated from increase in the subpopulations of the tumor cell state that are derived from T cell cluster, and similarly increase in the populations of the APC that are derived from T cell cluster. So this is how they use the bioinformatics and the publicly available data along with their RNA-seq data to come up to this point. which is also in line with the observations that are published in previous literatures. So I hope you understood how they clustered and characterized these tumors and APCs. If you need to understand more about this experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.